it will taper. After branching, it will rebranch like this. And these rebranches will also rebranch and it will form a tree-like structure, and it is involved in formation of synapse. A synapse is formed between two neurons. As I have already told, neurons are contiguous. These are not continuous. These are not in physical contact to each other, but functionally these are attached to each other through synapses. So this is dendrite, and it is having MAP. MAP is seen in only dendrite. It is microtubule associated protein. So MAP is seen only in dendrites, not seen in axons, especially microtubule associated protein type of 2. So a typical dendrite is having nasal substances. It is having microtubule associated protein, and it is having microfibrils, neurofibrils, and neurofilaments. On the other hand, there is a single large process known as axon. Axon is efferent bringing impulse from the soma to the distal part or terminal part of the axon, and it is involved in formation of synapse in its terminal part. So axon is covered by axolemma. It is having a outer covering of membrane known as axolemma, and it is having initially part which is clear zone. It is not having nasal substances, and it is dilated part of the neuron known as axon hillock. The part adjacent to the axon hillock and free from myelin sheath known as initial segment. Initial segment is involved in generation of the action potential or impulse and involved in propagation of the impulse from soma to the distal part of the axon. This axon covered by axolemma and having axoplasm inside, and it is having also branches which arises at right angle to the axon, but it maintains its diameter. The terminal part of the axon is having branching and small terminal branches known as telodendria. These are telodendria, and these are involved in formation of synapses. So afferent impulses coming toward the soma from the dendrites, and efferent are leaving the soma through the axon to the terminal part of the axon. It forms synapses. It may be in form of axodendrite. It may be in form of axonic. It may be in form of muscle neuromuscular junction or neuroglandular junction in case of lower motor neuron, or it may form axodendrite or axosomatic synapse which is inhibitory in nature. So the terminal part of the axon which is having telodendria involved in formation of synapses with other neurons or efferent organs, or it may form receptor in case of sensory neuron. So what is a myelin sheath? Or myelination? The myelination of the axon, only axons are having a sheath of lipoproteinous substance known as myelin are covered by this sheath, only axons. Neither soma nor dendrites are covered by myelin sheath. What is myelin sheath and how it is formed? It is a sheath, can be seen around the axon. It is a sheath its short intervals about 1 to 3 millimeters in case of tracts and the peripheral nerves. So these are segments of the axon covered by a lipoproteinous substance known as myelin sheath, and process of formation of the myelin sheath is known as myelination. So how myelin sheath is formed around the neurons and tracts? The myelination in central nervous system is done by oligodendroglia or oligodendrocytes. In case of peripheral nervous system, myelination is done by Schwann cells. One Schwann cell is dedicated to the single neurofiber or the axon, and it myelinates only single or one axon. In case of central nervous system, one oligodendrocyte or oligodendroglia myelinate many axons. Let me explain how the myelin sheath is formed. Here is the axon, and here is the Schwann cell. Initially, these are two different cells. This is part of axon and this is independent Schwann cells or oligodendrocyte. In later part, this will be axon and there will be a dent on the Schwann cell. In next stage, this axon will be engulfed by this Schwann cell like this. And here is the nuclei of the Schwann cell. So in initial stage of myelination, there is axon and there is Schwann cell, axon and Schwann cell. This Schwann cell will encircle or engulf the axon. Now this is axon, nucleus of the axon. This is Schwann cell, nucleus of Schwann cell. Here is the axon. This part which is connecting the outer environment to the axon and bilate, this part of Schwann cell is known as mesoaxon. Mesoaxon. This is Schwann cell, and this is axon. Now, this mesoaxon winds round the axon. This mesoaxon winds round this axon, and it will encircle this axon and forming a concentric layer of the mesoaxon around the axon like this. We have seen axon. Here is axon, and here is mesoaxon. It will encircle the axon, and here is so in later part of myelination there is axon, here is mesoaxon. This mesoaxon will encircle the axon to form a concentric layer of the mesoaxon, and here is nucleus of the Schwann cell. So these multiple layers, these concentric layers surrounding the axon will deposit a lipoproteinous substance known as myelin, which is deposited in the multiple layers of the plasma membrane of the Schwann cell. So this is myelin sheath formed by the membrane or the exolemma and the lipoproteinous substance known as myelin. So this is myelin sheath, and outermost covering of the myelin sheath, which is formed by the cytoplasm of the Schwann cell, and nucleus is inside this cytoplasm, this outermost layer. Outermost layer of the myelin sheath is formed by the cytoplasm and the cell membrane of the Schwann cell having nuclei. This is known as neurolemma or neurolemma, and this is known as myelin sheath. So what is neurolemma and what is myelin sheath? And this is axon. Axon surrounded by this myelin sheath is myelination, and outermost is neurolemma. In case of non-myelinated axons, in case of non-myelinated axons in peripheral nervous system, the axon is covered by a single layer of Schwann cell cytoplasm, and it will sheath many neurons. This sheath formed around the non-myelinated axons in peripheral nervous system. So neurons not having myelin sheath are having neurolemma or neurolemma, and this forms a basis for regeneration of the peripheral nervous system after some injury, some toxins or some poisonous substances. The distal part which is degenerated, like in Wollerin degeneration, this is regenerated by this sheath of Schwann cell known as neurulima. So part of CNS which is injured is not repaired, but part of peripheral nervous system which is injured is repaired due to presence of neurulima formed by Schwann cells. In case of oligodendrocytes in central nervous system, it forms the myelin sheath, but it doesn't form a sheath of cytoplasm and cell membrane on non-myelinated axons. So central nervous system when injured is not regenerated, and it forms a scar tissue by astrocytes in central nervous system. So up to now we have seen what is a neuron. A neuron is genetic, anatomical, functional and trophic unit of the nervous system. Cells lost the capacity to divide and form the other cells. So cells of nervous system or neurons are non-divisible cells, and these cells lost the property of cell division. Cells having a property of perceive the stimuli from internal and external environment, and having the property to generate action potential and propagate the action potential unattenuated for a long distance, and these cells are having capacity to receive the impulse from outer environment and transmit to the central nervous system, that is highest center of consciousness, that is cerebrum, and bringing the motor command towards the muscles and the glands. So this was the structure of the neuron, and this is the process of formation of the myelin sheath in case of axons. So this is formation of myelination. Now what is white matter and what is grey matter? Here you have seen the myelination, you have seen the cell body or soma, and you have seen the small and arborized processes known as dendrites. Myelin is formed by a combination of lipids and proteins. This myelin gives a typical appearance of white color in living person, or it looks pale in a dead person. So this whitish or pale color, which is seen due to formation of myelin sheath, this whitish part of brain which is having axons, and part of peripheral nerve system which is having very large axons in form of spinal nerves and cranial nerves. But always remember, white matter and grey matter are applicable for only central nervous system. These terms are not used for the peripheral nervous system. Nerves are also having myelin sheath. These also give appearance of white, and ganglia are collections of cell bodies are grey, but these are not called as grey and white matter. So we have seen the myelination, white appearance of myelination, and the grey appearance of the cell body and the dendrites, because these are not myelinated. So what is grey matter and what is white matter? Here grey matter and here is white matter. So we have seen Gray matter is collection of cell, bodies or perikaryones or somas like this. Here I will explain the gray matter and white matter. For examples, here is a tree having multiple branches. I am using this tree for the central nervous system, that is brain and spinal cord, and on this branch of tree there are many monkeys are sitting here having ear and mouth and other parts of their body. These multiple or many monkeys are sitting on the same branch of the tree, and these monkeys are having long tails. These are the tails of these monkeys. So the collection or group of these monkeys is gray matter because it is formed by these perikaryones or soma or cell bodies, and these tails collectively form the collection of axons known as fasciculus. The individual tail of a monkey is a single axon or neurofiber, and this is collection is known as fasciculus. Here I will explain one more thing. This cell, this single axon is covered by axolemma, myelin sheath, and then it is covered by neurolemma. So single axon is covered by axolemma, myelin sheath and neurolemma. Then it is covered by a sheath of connective tissue known as endoneurium. So it is covered by axolemma, then myelin, then neurolemma, and then endoneurium. This axon, now it will form a fasciculus which is having multiple axons, and these axons are covered by a sheath of connective tissue known as perineurium, and multiple fasciculus will form the trunk of a nerve or a bundle of the fasciculus. This is known as nerve or trunk, and this is covered by epineurium. So we have seen the coverings of a single nerve fiber or axon: axolemma, myelin sheath, neurolemma, endoneurium, perineurium, and epineurium. So parts of central nervous system, that is brain and spinal cord, are protected by the cranial cavity and the vertebral cavity. In case of peripheral nervous system, the nerve is protected by this axolemma, this myelin sheath, neurolemma, endoneurium, perineurium, and epineurium. Besides these, it is also protected by the covering fascia, the muscles, the tendons, and the organ which is innervated by this muscle, this nerve. So we have seen what is soma, what is gray matter, what is white matter. Here I want to explain some more things related to the gray matter and white matter. When this collection of these somas or this collection of nerve cell bodies outside the nervous system, that is outside the central nervous system. It is known as ganglion. When it is outside the central nervous system, this is known as ganglia. When this collection of the cell bodies or perikaryones it's inside the central nervous system, this is called as nuclei. This is called as nuclei. This collection of the axons or tails of monkeys, when it is seen inside the central nervous system, it is known as tract in case of vertical column, and in peripheral nervous system it is known as the peripheral nerve. Either it may be cranial nerve, it may be spinal nerve, or it may be autonomic nerve. So what is gray and white matter? When these fibers are in vertical column inside the CNS, these are known as tracts, like ascending and descending tracts of the spinal cord. When these are seen in case of brain, these are seen in form of three types of fibers, that is association fiber, commissure fiber, and there are three types of fibers. These are association fibers, when these connect the same cerebral hemisphere, these are known as cerebral association fiber. When these connect the right and left part of the brain, mainly when these connect the right and left cerebral hemisphere, these are known as commissure fibers. The anterior commissure connected insula to the anterior pole of the temporal lobe, and the commissure fiber which is seen in brain, large commissure known as corpus callosum. And third type of fibers are known as projection fibers. These fibers connect the one part of the brain to the other part, like from cerebrum to the brainstem, and from cerebrum to the cerebellum, from the cerebral hemisphere to the spinal cord in form of tracts. These are known as projection fibers. So white matter in case of central nervous system can be seen in form of these association fibers, commissure fiber, and projection fibers. The gray matter inside the central nervous system can be seen in form of the nuclei and also seen in form of cortices. What are cortices? In case of superior colliculi, cerebellum and cerebrum, the outermost part of the cerebrum, cerebellum and superior colliculi are covered by a sheath of gray matter. This sheath is known as cortex or cortices. This cortex is having horizontal arrangement of these perikaryones or gray matter. Anatomically, these are horizontal, but functionally these are vertical columns of the gray matter. These are known as cortex, like cerebral cortex, cerebellar cortex, or cortex of the superior colliculi of the midbrain. So arrangement of the gray matter fibers inside the CNS in form of the nuclei, in form of cortex. We can say nuclei like basal nuclei. Previously it was known as basal ganglion. Nowadays it is known as basal nuclei. The nuclei of cranial nerves in brainstem are also examples of gray matter inside the CNS. And I have already told cortex, cortices. In case of cerebrum, it is cerebral cortex. In case of cerebellum, it is cerebellar cortex. And it is in case of superior colliculi, this is cortex of superior colliculi. And in peripheral nervous system, collection of the white matter is in form of nerves, spinal nerve, cranial nerve, or the autonomic nerves. In case of the gray matter in peripheral nervous system, these are known as ganglia. There are sensory ganglia. These can be seen in form of dorsal root ganglion of the spinal nerves and cranial nerve ganglion of 5th, 7th, 8th, 9th and 10th cranial nerves, and it is seen in the sympathetic and parasympathetic ganglion of the peripheral nervous system. So ganglion in form of the dorsal root ganglion, the cranial root ganglion or cranial nerve ganglion, and autonomic ganglion. The dorsal root ganglion and cranial root ganglion are having pseudo-unipolar types of neurons. Only 0.5% of neurons in the total nervous system are pseudo-unipolar, and these are having a single convoluted process from soma, which will divide in a peripheral process and intercentral process. The peripheral process and central process anatomically both are axons, but functionally one process, that is peripheral process, acts as a dendrite and central process acts as an axon. So these collections of gray matter outside the central nervous system are known as dorsal root ganglion and cranial root ganglion. These are sensory ganglion, and these are having pseudo-unipolar. Nowadays these are known as unipolar neurons. These are having a peripheral and a central processes. The peripheral process, which is bringing the impulse from the periphery to the central process without relay in the soma. It acts as dendrite, and the central process, which is bringing impulse from the periphery to the central nervous system, acts as an axon. Autonomic ganglia are motor ganglia. These can be seen in form of sympathetic ganglia on both sides of the spinal cord in form of sympathetic chain. These are paravertebral ganglia. There are some prevertebral ganglia which are having preganglion fibers from the subsplanchnic nerves. Greater, lesser and least subsplanchnic nerve form the celiac, superior mesenteric and inferior mesenteric ganglia of the prevertebral type of ganglion. And the parasympathetic ganglia relay near the effector organ, and these are visceromotor, and these are having multiple ganglia. I have already told, like pterygopalatine, submandibular, otic and ciliary ganglion in the head and neck, and multiple ganglia in the thorax and abdomen to supply the viscera of the thorax and abdomen. So we have seen in this lecture this structure of the neuron, the formation of myelin. Myelin acts as a insulator, and it provides nutritional and structural and functional support to the axon, and it also plays a great role in transmission or propagation of the nerve impulse, which I will talk in my next lecture. What is saltatory conduction and how it speeds the conduction of impulse in the nerve or tract, and what is gray matter and what is white matter, what is ganglia, what is nuclei.